Welcome to ON A-Level Science. So today I'm going to talk about topical questions from chapter 3 Enzymes of AS level biology. So hopefully this helps. I'm going to talk about the different types of graphs which may pop up in your test and the different types of MCQ questions of paper 1. So let's look at number 1. An enzyme is completely denatured at 50 degrees Celsius. A fixed concentration of this enzyme is added to a fixed concentration of a substrate. Okay, the time taken for completion of this reaction is measured at different temperature. Which graph shows the result? Okay, now it says this enzyme is denatured at 50 degrees Celsius. So here it says time taken for completion of reaction. So that's the y-axis for every graph here. So at 50 degrees Celsius, since it's denatured, so the time taken for completion of this reaction would be it will be infinite because the reaction would not proceed. So for each of these graph, as you can see, if we draw a line at 50 degrees Celsius, we are going to see that the reaction is still occurring. Even here at 50 degrees Celsius, some activity can even be seen. That means that the reaction is still completed at 50. Also at T, still some reaction is taking place at 50 and the reaction is being completed. Only at this option, if we draw a line at 50 degrees Celsius, sorry, that's a very curved line, not also a straight line. Anyways, so if we draw a line I'm really sorry for the okay, so if we draw a straight line at 50 degrees Celsius, we can see that it does not intersect with any point in the graph. So this shows that the enzyme is indeed denatured and the reaction will not get completed. So as a result, the answer to our question 1 is C. So this is a bit technical question, but once you know how to figure out, these are pretty simple. Okay, question number 2. Which properties are characteristics of a non-competitive inhibitor of an enzyme? So non-competitive inhibitor, as we know, it does not bind with the active site, it does not compete with the substrate, it binds with another site of the enzyme. Okay, so that another site can be the allosteric site for some who may know it, or it's a different place than the active site. So the first A and B can be easily eliminated. Now, effect of adding more substrate does not have any effect on a non-competitive inhibitor. It has an effect for competitive inhibitor. So competitive inhibitors bind with the substrate, where if you increase the substrate concentration, then it reduces the inhibition. That's for competitive, but not for non-competitive. As it's non-competitive, it will have no effect on inhibition if you increase the substrate concentration. Okay, so question number 3. It says a piece of mammalian tissue was homogenized and subjected to different centrifugation yield for 4 subcellular fractions. Okay, the activity within each fraction of 4 different types of enzymes ABCD were investigated. Each bar chart shows the results of investigation of hydrolytic enzyme activity. So here we can see the key where they have given us the cell organelles which they have investigated with. So first there is nuclei, then there is mitochondria, lysosomes and ribosomes. So we need to focus here on this word, hydrolytic enzymes. Okay, so hydrolytic enzyme, as you know, are mainly present in the lysosomes. So over here the y-axis is the activity, so the activity shall be undoubtedly highest in the lysosomes. So if you look at option A, the activity is not highest in the lysosome, it's shown highest in mitochondria, so it's wrong. So in B, lysosome is showing a very low activity, which is wrong. In C, only in C, you can see the highest activity in lysosome, and in D it shows very low activity, so undoubtedly the answer should be C. As for mitochondria, there are enzymes which are required for respiration process to take place, so there is some activity. Then there is ribosomes and nucleus, so it's just mainly the lysosomes which is the main focus of this question. Okay, So in question number 4, let's see. A metabolic pathway is, so here we can see a metabolic pathway. It shows a reactant, enzyme 1, substance X, enzyme 2, substance Y, enzyme 3, and end product. Okay, so what would be the effect of adding a small amount of non-competitive inhibitor on enzyme 2? Okay, so on enzyme 2 we are going to add a non-competitive inhibitor. Okay, so what will be the effect of adding non-competitive inhibitor on enzyme 2? That's the question they're asking us. So it's pretty simple. So if there is a non-competitive inhibitor added here, that means there will be less of substance X being there will be more substance X remaining, because less substance X will get converted, so X will increase in concentration. So this can only be seen in option B, but let's still read the other options. So option A says enzyme 2 will be partially denatured. No, it's not going to denature. It's not going to partially denature. Then it says option C, substance X would no longer be formed. No, substance Y will no longer be formed. That's wrong, because still some amount of substance Y will still be formed, but it will be in low concentration, because not all the enzymes are denatured. Not all the enzymes stop working because of the non-competitive inhibitors. Some amount of enzymes will still function and substance Y will still be formed. The initial reaction will no longer be metabolized. No, that's wrong. Okay, so that's for question number 4. Let's see question number 5, another graph question. So there are a lot of graph questions which you guys may encounter from this chapter, enzymes. Okay, so 5 says the initial rate of reaction catalyzed when enzyme was measured at various substrate concentration. Okay, so the x-axis are all substrate concentrations. Which graph shows the effect of a low concentration of non-competitive inhibitor on the reaction? Okay, it says low concentration of non-competitive inhibitor. So here they have given the key. It says uninhibited reaction and inhibited reaction. Okay, so these dotted lines are uninhibited reactions and these full lines are the inhibited reactions. So it says which has a low concentration of non-competitive inhibitor on the reaction. Okay, now let's take a look at the graphs. So these, we know this is a very general trend. For non-competitive inhibitor, the graph always looks like this. Okay, so this is obviously the inhibited reaction. So inhibited reaction, the graph will not reach its Vmax. Okay, it will be at a lower rate because we are adding a non-competitive inhibitor. That's a trend for any non-competitive inhibitor graph. And uninhibited reaction, it will be as usual enzyme activity. That means it will reach its Vmax and it will look like this. So this is